Hi, everyone, and thank you to the NBSTRN for having me today and for allowing the Immunology Working Group to present our work to you guys. So I'm just going to go ahead and get started. So to begin, I wanted to talk about the overview of Clingen and our Severe Combined Immunodeficiency Expert Panels. And you may be wondering, what is Clingen? So it stands for Clinical Genome Resource and does exactly as the name says. It's an NIH-funded resource that defines clinical relevance of both genes and variants for the use in precision medicine as well as research. And so this is a map of Clingen members that partake in the working groups. So countries with members are shown in blue. Across 46 countries and territories, there are currently 1,897 total contributors to Clingen's efforts. So as you can tell, Clingen is working with experts all over the world currently. And so all of these members share the same mission of building a genomic knowledge-based improved patient care. And this includes the supporting key goals of sharing genome and phenotypic data through a federated database for clinical research and use, which therefore serves as a centralized location where clinicians and researchers can aggregate data regarding genes, genetic conditions, and the genetic variants that cause them. Another goal is developing and implementing a standardized process for both annotation of literature and also interpretation of genes and variants. We're also developing data and software infrastructure, as well as computational processes that will help curation proceed at a larger scale. And also we have a hope to integrate this into the healthcare setting. Another goal is to enhance and accelerate expert review of the clinical relevance of different genes and variants. So this is done by bringing in experts together in the same group setting to discuss discordant interpretations regarding genes and variants. And I'm going to talk about this a little bit more as we talk about the different groups that you can see in Clingen in the upcoming slide. And lastly, another goal of Clingen is to disseminate and integrate all knowledge and resources of Clingen to the broader community for the betterment of patient care, which, as you can see, goes with our overall mission to the left. So now I want to talk a little bit about the different nomenclature found in curation settings at Clingen. First, we have that clinical domain working group. This is an overarching term that includes all members participating in activities. So for example, the SCID variant and gene curation exists as two of multiple different subgroups of the immunology working group. And so you'll see this displayed in a figure on the next slide. Next, we have the Variant Curation Expert Panel, also more commonly known as a Vsep. This is a subgroup of the immunology working group that acts to first specify rules for variant curation for a specific subset of gene or gene families. So for us, that would be SCID, and then acts to curate these variants where they then go to ClinVar and they'll receive a 3-star submission status in ClinVar. And then finally, we have the Gene Disease Curation Expert Panel, also known as a GCEP. And this is a subgroup that works to focus on Clingen gene validity curation and determine the association between a particular gene and disease. So here's a visual of immunology clinical working group. And at the top, we have the leadership positions, which include the co-chairs of the different subgroups, as well as the Clingen liaison and the senior coordinators. The executive committee, if you look a little bit lower, is made up of experts on a particular phenotypic subgroup. So for us, we have experts in the hereditary angioedemas, for SCID, and for antibody deficiencies. And this is all with an overall overarching background. All the experts have a strong background in immunology as well as those specific areas of expertise. So now that I have talked a little bit about the mission and the structure of Clingen, I want to just give a little brief review of what SCID actually is. So SCID stands for Severe Combined Immunodeficiency. For reference, some of you may be familiar with the case of David Vetter, more famously known as Bubble Boy, due to living in the confines of a small plastic bubble. And David Vetter had a diagnosis of SCID. SCID has an incidence of 1 in 100,000 births and is caused by lack of T cells at birth and has a high mortality from inability of these patients to fight off infections. So typically those with SCID do not survive longer than about 1 to 2 years of age without a form of immune restoring treatment. And possible treatments for this include a stem cell transplant or a possible gene therapy. And the decision of which treatment is used is more informed with a known genetic etiology. And I will discuss more about this later. And in an effort to help diagnose this disease, SCID was officially placed on the newborn screen in all 50 states at the end of 2019. So let's talk about how the newborn screening method for SCID works. As many of you know, newborns get a heel prick at birth twice, in which a dried blood spot is tested for a variety of different conditions. And for SCID, this is looking at TREX. So TREX are our T cell receptor excision circles. And so normally when our cells are making the T cell receptors, the T cell receptor locus, which is shown to the right, splices out the central portion of that locus and it ligates or glues itself back together and forms the T cell receptor. The portion of the locus that is cut out actually conjoins in on itself and forms a circle that we call the T cell receptor excision circle, otherwise known as TREX. So if a baby is making T cell receptors, we would expect to see high level of TREX. If a baby's not making T cell receptors, we would expect to see low TREX. And so this is, just a reminder, a screen. It is not diagnostic. So what happens when we get low TREX? Well, there are multiple different causes for this. A newborn could have SCID, another immunodeficiency like combined immunodeficiency, otherwise known as CID. They could have a chromosomal abnormality. It could be caused by prematurity or secondary losses as well. And it is vital that we confirm the diagnosis for low TREX as soon as possible. And this is because if it actually turns out to be SCID, treatment needs to occur abruptly. And this is why we consider it a medical emergency. But why is it a medical emergency? So SCID is treated urgently because treatment needs to occur as soon as possible. And it has been shown in different studies that there's a higher probability of survival if treated before infection occurs and also before 3.5 months of age. Conversely, the probability of survival is lower if there is an active infection by the time of transplantation or if it occurs after 3.5 months of age. And you can kind of see the probability of survival differs in that figure to the right, with those with a transplantation before 3.5 age having a way higher probability of survival than those having a transplantation after 3.5 months of age. Similarly, if you have an active infection at the time of transplant, the probability of survival is lower rather than having no infection at the time of transplantation. It's understood that a swift diagnosis is vital to the survival of those with SCID. The best way to diagnose SCID is molecularly, and this is where Clingen and the SCID variant and Gene CurationExpert Panels come into the picture. So let's first start with the GCEP, or the Gene CurationExpert Panel. And that is highlighted in this figure of the different subgroups of the immunology working group. So the Gene Curation Working Group, similar in structure to the Immunology Working Group, first developed a framework for how GCEPs are supposed to work. So a GCEP is to assess the strength of evidence supporting a gene disease association. And this framework defines criteria needed to demonstrate causality, organizes and quantifies different types of evidence, and allows curators to methodically assign clinical validity classification. And so the framework provides a semi-quantitative measurement for the strength of evidence of a gene-disease relationship that correlates to a qualitative classification. So for example, definitive, strong, moderate, limited, no reported evidence, or conflicting evidence. And within the GCEP, the classifications derived with this framework are reviewed and confirmed or adjusted based on the clinical expertise of appropriate disease experts. And this evidence-based systematic method to assess the strength of gene-disease relationship will facilitate more knowledgeable utilization of genomic variants in clinical and research settings. And if you're interested in learning more about this specific framework that was created, I've attached the paper by Strand et al. Here, but also it will be found in my resources as well. And so the SCID CID GCEP, so SCID for severe combined immunodeficiency and combined immunodeficiency for CID, is currently working on providing gene disease association classifications for 120 different genes that are associated with both severe combined immunodeficiency as well as combined immunodeficiency. And these genes are derived from tables 1 and 2 of the Human Inborn Errors of Immunity classifications from the International Union of Immunological Societies Expert Committee, or the IUIS. And I've listed some of the genes in this table below, but for a full list, you can visit the IUIS website or see my resources at the end. Of note, genes for SCID are marked with an asterisk. And so far, we have 37 genes that are published and curated. And so next, we're going to move on to the Variant Curation Expert Panel. Here you can see this subgroup highlighted in the immunology working group, just for reference of the structure of Clingen. So traditionally, a Vsep is a team across multiple institutions that's comprised of gene experts and biocurators who function to determine the pathogenicity of genetic variants. And so the basis for classification is founded on ACMG guidelines found in the paper you see below by Richards et al. But these, what we call specifications for variant classification, are also modified to fit the specific nuances of the genes with help of experts and biocurators. So once these specifications are adjusted, they're then approved by the Clingen SVI and Variant Curation Working Group, which is a different working group. And curation of variants can begin to test these specifications that were created by our Vsep. So the point of such a rigorous process to create guidelines, or otherwise known as specifications for variant classification, is to improve the quality of our assertions and in turn increase the number of genetic variants in ClinVar that receive a 3-star submission. So moving on to the SCID Vsep specifically, we were approved on January 28th of 2021. We meet one time a month with the goal of creating guidelines and ultimately curating variants across 19 different SCID genes. These genes are also found in Table 1 of the IUIS document that I previously referred to. And so this is a figure that shows the current phase of the Vsep. We've created specifications or guidelines for variant classifications for 9 different genes. That's been submitted, and we've received some initial feedback. We've edited that feedback, and now we are in the pilot phase in which we have to test our specifications across 90 different variants. So the point of this is to see how our guidelines for classifications work on different types of variants, including those that are suspected to be pathogenic, some that are suspected to be benign, and some that are currently unknown. So once we complete our pilot phase and submit back, we receive hopefully final approval in step 4, at which point we can move on to sustained curation of variants related to SCID genes. So why is the SCID Vsep and GCEP so important? Currently, there are many uncurated variants or variants of uncertain significance related to SCID in ClinVar. And this can have really serious clinical implications. When clinicians order a test, they may bypass a VUS, thereby preventing official diagnosis of SCID for a baby that could benefit from a stem cell transplant. Conversely, clinical diagnosis of SCID without a confirmed molecular diagnosis may pose harm to the patient, as we know that sometimes T-cell lymphopenia can resolve, and a stem cell transplant is too drastic of a measure for that patient. Alternatively, some molecular causes of SCID show evidence that certain types of radiation or chemo treatments are contraindicated. So having a molecular diagnosis can help determine what treatment is necessary for a diagnosis of SCID that goes with a particular gene or variant. And so this is why the work of the GCEP and the Vsep are so important in relation to the SCID newborn screen, is because it helps us increase diagnostic yield, increases the probability of survival for patients in SCID, and also improves clinical validity. So in summary, the vision of the NBS-TRN is to identify, develop, and test promising new screening technologies, increase specificity of newborn screening while also expanding the number of conditions tested, and to develop experimental treatments and disease management strategies for these diseases. And so our work on the SCID GCEP and Vseps helps the NBS-TRN in all of these goals by validating the proposed genetic etiologies of the abnormal newborn screen by either diagnosing or excluding SCID. We are increasing the specificity of the newborn screen. Furthermore, by classifying the abundant number of variants of uncertain significance as pathogenic or not pathogenic, we drastically improve the diagnostic yield of the newborn screen. And lastly, the NBS-TRN seeks to develop experimental treatment or to improve disease management. And with Clingen, Vsep, and GCEPs pinpointing a genetic cause for SCID, patients can receive the most appropriate stem cell transplant as indicated by their genetic etiology. And this overall plays a vital role in patient survival. And so that's kind of the summary of the overarching function of what we are doing in the variant and gene curation panels right now. If you have any questions, please do not hesitate to reach out to me, or you can ask them live. But I do want to thank the Clingen Immunology Working Group for their contributions thus far to our curation efforts. And also thank you again for having me today and allowing me to share the work of our Immunology Working Group thus far.